off because there is some issue with my camera. That's why I'm not able to switch on it. Okay? Chalo. We'll start the class. As we all know, this is our first lecture. In first lecture, we will start with our new topic. That's your glycolysis. We'll start with glycolysis. So firstly, we will start with respiration and its types, and then glycolysis, and then link reaction. What is the weightage of this chapter? If you have look into this respiration chapter in 2023, 2 questions is already asked. In 2021, 3 questions asked. 2021, 2 questions. 20 again 2 questions, and 29 again 3 questions is already asked. So it means this chapter has also a quite high weightage. Okay, then we'll start with quick recap of previous chapters. Okay, guys, did you study the cell? Just give me a second. Yeah, you have already studied the chapter cell, the unit of life, right or not? आप लोग पहले से ही सेल द यूनिट ऑफ लाइफ चैप्टर पढ़ चुके हैं. And in this in cell the unit of life chapter, you have studied about cytoplasm, mitochondria, and the third one cell organelle is chloroplast. Why I'm going to recap it here, because all these cell organelles, chloroplast, yeah, all these cell organelles, we have 2 cell organelles here, mitochondria and chloroplast. We will check in the respiration chapter. There is quite role of cytoplasm, and there is also an important role of mitochondria. In the simple language, we also say mitochondria. What is cytoplasm? Cytoplasm is the main arena for cellular activities, no?
Yes, ma'am.
Right, Harshada?
Yes, ma'am.
इसी को हम मेन एरेना बोलते हैं. मतलब हमारे सेल का मैक्सिमम से मैक्सिमम जो एक्टिविटीज होता है वो साइटोप्लाज्म के अंदर चलता है. एंड इन द साइटोप्लाज्म वी हैव अ सेल ऑर्गेनेली व्हिच इज माइटोकॉन्ड्रिया, एंड वी ऑल नो माइटोकॉन्ड्रिया इज आल्सो नोन एज
Powerhouse of the cell.
powerhouse of the cell. So in the next chapter, basically photosynthesis of in higher blood, we will study how mitochondria is able to produce energy in the form of
ATP.
ATP. What is the full form of ATP?
Adenosine triphosphate.
Very good. Adenosine triphosphate. So in the next chapter, we will study how we are going to produce ATP. Why we are calling mitochondria as the powerhouse of the cell. And in the previous chapter, we have also studied mitochondria is a double membranous cell organelle. Double membranous hota hai na? And in the inner membrane has foldings, and these foldings, or we can say these invaginations, are known as crista. And we have also studied mitochondria is also known as site of aerobic respiration.
Yes, ma'am.
It is the site of aerobic respiration, na? And what is chloroplast? What is chloroplast?
Green pigments.
We call it kitchen of the cell. Why we call it kitchen of the cell? Because it's help to produce food. It catalyzes which process? It catalyzes which process?
Photosynthesis.
Photosynthesis. And in the cell chapter, we have studied the basic structure of a chloroplast. Chloroplast is a double membranous organelle. Right? And in the chloroplast, we have some coin-like structures stalk over each other, just like this. And these stalk-like structures are known as?
Thylakoids.
One structure is known as thylakoid. And the entire stalk is known as? Grana. And 2 granas are connected by are joined by a connection. That connection is known as
Stroma.
Stromal,
Lamellae.
lamella, very good. And the outer environment is known as
Ribosomes.
stroma. Outer environment is known as stroma, not ribosome. Ribosomes are the protein factories, na?
Yes, ma'am.
Ribosomes are protein factories. Yes, Arshida. So this is the basic data that you should know before starting the plant physiology. Okay? So let's start with the respiration in plant. So can you just tell me the basic definition of respiration? What basically respiration term signify?
The breakdown of oxygen to produce the breakdown of food in the presence of oxygen.
Anyone else?
The inhale of O2 and the exhale of CO2 is called expiration.
The
Respiration is the
process of breathing in oxygen and breathing out carbon dioxide.
have two process. The first one is respiration and the second one is breathing. Differentiate between these two terms. Breathing is the exchange of gases. Exchange of gases means inhale करेंगे which gas
Oxygen. O2
and
oxygen.
exhale
CO2
CO2.
carbon dioxide.
CO2. ठीक है? We are inhaling this oxygen and this oxygen will go into the cells. This oxygen will go into the cell and by the help of this oxygen, in our cell will do oxidation of food. Oxidation of food is known as respiration. Oxidation of food is known as respiration. Food is like glucose na C6H12O6. In the presence of oxygen, it get break down into CO2 plus H2O plus
Energy.
energy and
ATP.
ATP. And what happens in photosynthesis? Can anyone tell me the equation of photosynthesis? Obviously, CO2 plus H2O. In the presence of
Light, it gives C6H12O6 plus H
chlorophyll
and
and
oxygen.
light is used to give us C6H12O6
And
plus
water.
O2 plus
Water.
water. Very nice. So by the process of photosynthesis, we are making
Glucose.
glucose and this glucose is getting break down into cell with the help of
Oxygen.
Oxygen.
oxygen. This
Oxygen.
process is known as
Respiration.
Respiration.
respiration. So basically why we are doing photosynthesis, we are doing basically plants. Okay, plants are doing photosynthesis just to gain energy na. In the photosynthesis they are not gaining energy, they will gain energy after respiration. So respiration is the oxidation of food where within the cell. So respiration is a which type of process, catabolism or anabolism?
Catabolism.
Catabolism. In the same time, respiration is also known as physiochemical process. Also known as physiochemical process in which we are doing breaking down of carbon-carbon bond. कार्बन बॉन्ड ब्रेक हो रहा है ना यहाँ पे. तो इन दिस चैप्टर वी विल बेसिकली स्टडी कि हाउ वी आर कन्वर्टिंग दिस ग्लूकोज into CO2, H2O, and how many ATP we will gain. How many ATP we will synthesize. And this is not a single step process. We are writing it here in a single step. Up to the 10th class, we have just studied the single equation. But in higher classes, we'll study this is not a single step process. This is a very slow process and it is completely coordinated by enzymes. Completely coordinated by enzymes. Are you getting my point?
Yes, ma'am.
Yes,
So
ma'am.
what we have done, respiration. What is respiration? Respiration is the oxidation of food within the cell. Why we are doing oxidation? Because by oxidation of food, we will get ATP. And from where we are making this food? Food is making by the process of, we are synthesizing food by the process of photosynthesis.
Photosynthesis.
Done?
Yes, ma'am.
Okay then. Now we will study the simple steps. I'll tell you firstly, I'll tell you a flow chart. Firstly, we will convert this glucose into pyruvic acid. We will convert this glucose into pyruvic acid. Glucose is how many carbon molecule?
6.
6
6,000.
carbon.
It's
And
carbon.
pyruvic acid is a 3 carbon molecule. Then how many molecules of pyruvic acid will synthesize here? 2 molecules, na? Because pyruvic acid is a 3 carbon compound. Then process is known as glycolysis, which is a 10-step process. And what is the site of glycolysis? Glycolysis is carrying out in cytoplasm. साइटोप्लाज्म के अंदर ये प्रोसेस हो रहा है. देन आफ्टर मेकिंग पायरुविक एसिड इन द साइटोप्लाज्म, पायरुविक एसिड विल मूव टू द मैट्रिक्स ऑफ माइटोकॉन्ड्रिया. मैट्रिक्स ऑफ माइटोकॉन्ड्रिया के अंदर ये आपका मूव कर जाएगा, वेयर इट विल कन्वर्ट इनटू एसिटाइल कोए. एंड दिस रिएक्शन इज नोन एज conversion of pyruvic acid into acetyl CoA. This reaction is known as link reaction. After converting into acetyl CoA will combine with a molecule oxaloacetic acid, and then it will create CO2 plus H2. This step is known as which step? This is known as which step? Krebs cycle. Very good. Krebs cycle. So in this chapter, we are going to study these 3 steps. The first one is glycolysis, which is a 10-step process. Then we will study about link reaction, and then we will go into Krebs cycle. After finishing the Krebs cycle, I'll gain CO2, H2O, and some amount of
ATP.
ATP. Basically, this is our ultimate goal, na? We need to convert this glucose into CO2 and H2O. So in the conversion of CO2 H2O, we need to convert glucose into pyruvic acid, then into acetyl CoA, and then into CO2 and H2O. Can we start with our cycle now?
Yes, ma'am.
Yes, ma'am.
Okay.
Yes, ma'am.
Sure. So let's start with our cycle, which is your glycolysis. I'll share my handwritten notes, बच्चों. आप लोगों के साथ मैं अपने हैंड रिटन नोट शेयर करूंगी, एंड इफ यू नीड एंटायर चैप्टर के हैंड रिटन नोट्स तो आपको मेरे ग्रुप में मैं आपको शेयर कर दूंगी, तो आप प्रॉपर नोट्स वहां से बना लेना. बेसिकली अभी क्लास में जस्ट ट्राई टू लर्न सम सिंपल थिंग्स जिसमें बच्चों को सबसे ज्यादा कंफ्यूजन दे यूज टू फील. ग्लाइकोलाइसिस is a Greek word which is made up of 2 terms. Glyc matlab glucose and lysis means breakdown. Basically what we are doing in this process, we are doing breaking down of glucose now. And this glycolysis is given by 3 scientists. EMP pathway is also known as. This cycle is also known as EMP pathway. What is the full form of EMP? Amidon Meyerhof Parnaz. Ambidon, Meyerhof, Bernard, all the things you will get in my handwritten notes. Okay? And the first rule of glycolysis is this. What is the site of glycolysis? Cytoplasm.
Cytoplasm.
And this is a universal process. This is a universal process. It means in every organism, in every organism, glycolysis will be seen. Either it can be a human being, either it can be a bacteria, it can be a virus, anything else. In every organism, glycolysis is a universal process. There will be no use of glucose. If there is no use of glucose, then there will be no anaerobic or aerobic type of organism. In anaerobic also, glycolysis will happen. Right or not? So let's start with the proper cycle. Okay? Firstly, we will have a glucose. Glucose is, you all know, na, C6H12O6.
Yes, ma'am.
Okay? Firstly, we will convert this glucose into which?
Pyruvate.
No, I already told you, na, glucose to conversion of glucose to pyruvic acid is a 10-step process. So I'm writing down here all the 10 steps. I'm writing down here all the 10 steps which are involved in between glucose to pyruvic acid. So glucose will convert into glucose 6
Phosphate.
phosphate. And then glucose 6 phosphate will get converted into
Fructose 6
Fructose phosphate.
phosphate.
fructose 6 phosphate. Fructose 6 phosphate will get converted into
Fructose 1, 6 phosphate.
1,6 bisphosphate. Achha, what is the difference between glucose and fructose? What is the difference between glucose and fructose?
Ma'am, in fructose there are two spaces to uh, add carbon.
To add phosphate. Yeah, we can also say this, but like if I told you glucose is a aldose compound and fructose is a ketose compound. Ketone
Glucose consists of
है
an
ना? हाँ. And glucose has only one free end where I have attached a phosphate group. So ये phosphate कहाँ से आएगा? From where this phosphate group is coming? We need to break down a ATP na. We will break down a single molecule of ATP into ATP is adenosine triphosphate na. It means it has 3 phosphate. So I am getting converted into ADP plus inorganic phosphate. ADP. What is the full form of ADP? Adenosine diphosphate. And this release phosphate will get attached to the glucose. And glucose की किस position पे attach हो जाएगा ये? किस position पे attach हो जाएगा? It will get attached to the at 6th position. It will get attached at the 6th position of glucose. And then we will convert this glucose 6 phosphate into fructose 6 phosphate. This is a normal step of isomerization na. Glucose 6 को fructose में convert कर दिया. And after converting glucose 6 to into glucose 1,6. यहाँ पे हमने क्या किया है? At 6th position we already have one phosphate. At 6th position we already have one phosphate. Then I have added one more phosphate here. So it means again I need to convert ATP into ADP plus inorganic phosphate. Then one more phosphate will get attached to the first position of fructose. Basic structure. इधर एक phosphate attach है, इधर एक phosphate attach है. 1, 2, 3, 4 and ये आपका पूरा 5, 6 membered ring बन जाता है ऐसे ही. आता ना? इधर CH2OH होता है तो इधर phosphate होता है 1, 2, 3, 4, 5 and एक इधर CH2OH होता है 6. यहाँ पे phosphate attach होता है. Biomolecules में you have made these structures. समझ आ रहा है?
Yes, ma'am.
ठीक है? तो it means in the first step, we have consumed a ATP molecule, and in the third step, we have also consumed a ATP molecule. So we have already consumed, because in the glycolysis process, our ultimate aim is to produce ATP. But in the initial steps, we are losing ATPs. हमको ATP बनाना था, हमको ATP यहाँ पे फॉर्मेशन करना था, बट फॉर्मेशन से पहले हमको ATP यहाँ पे कंज्यूम करना पड़ गया. तो अब यही हमारा फ्रक्टोज 1,6 बिस्फोस्फेट किस में कन्वर्ट हो जाएगा? हरिए बच्चों, बताओ जल्दी जल्दी से. टेल मी.
Triose phosphate में.
Very good. It will get convert into and then triose phosphate will get converted into
Triose biphosphate.
yeah, we can say triose biphosphate, but before converting it into triose biphosphate will get converted into, there is a reversible reaction. यहाँ पे एक reversible reaction होने वाला है. Then triose phosphate will get converted into, in which? Yes, Aditya. In the short, we call it dihap. This is dihap. What is dihap here? Dihydroxy acetone.
Phosphate. Acetone phosphate.
Phosphate.
है ना? This is a reversible reaction. ठीक है? This is a reversible reaction. So we can call it, which type of enzyme we'll use here? Isomerase enzyme we can use. We are making isomer of a triose phosphate. Dihap is the isomer of triose phosphate. And then this triose phosphate will get converted into which?
2 into triose biphosphate.
Phosphoglyceric acid. Triose biphosphate.
Students are saying two names. The first one is saying triose bisphosphate, and another one is saying diphosphoglyceric acid. सुना ना? बोल रहे ना कोई? Both are same terms. दोनों की दोनों ही आपकी exactly same है. आप कुछ भी लिख सकते हैं. इनका सिर्फ और सिर्फ nomenclature है यहाँ पे. तो I'm writing it here. 1,3 diphosphoglyceric acid. So this was our first step. This is our second step. This is third. This one is fourth. This one is fifth. This one is sixth step. Then 1,3-diphosphoglyceric acid will get converted into which? 3
3-phosphoglycerate.
glyceric
Phosphate
acid, or we can also
acid.
call it glycerate. Then 3-phosphoglyceric acid will get converted into
Phosphoglycerate.
2-phosphoglycerate.
I'm writing it here, glyceric acid. ठीक है? बात same है. Then 2-phosphoglyceric acid will get converted into
2-phosphoenol pyruvate.
2-phosphoenol pyruvate. And then 2-phosphoenol pyruvate will get converted into our final product. Our final product is
2
Pyruvate
pyruvate.
acid.
Pyruvate.
very nice. No, it's not necessary, Anu. It's not necessary to write all the two names. You can write it according to your choice. If you are feeling triose phosphate is easy, then you can remember it. If you feel 1,3-diphosphoglyceric acid is easy to remember, then you can do it. You can
Okay,
also
ma'am.
write it triose bisphosphate. The choice is yours.
Okay, ma'am. Thank you.
Okay. Okay. So we'll try to remember this table. Now we will try to remember this table. Firstly, start with glucose. So first step, what we have done? In the first step, what we have done? We are adding a glucose at 6-phosphate. So before adding a glucose, before adding a phosphate group, we need to break down a ATP. So we have converted glucose into glucose 6-phosphate. And in this step, we will use an enzyme. And the name of enzyme is hexokinase. This is very important process. Very important. नीट में क्वेश्चन बहुत बार पूछा जा चुका है. Which enzyme?
Hexokinase.
Hexokinase. ठीक है, will convert glucose into glucose 6-phosphate with the help of hexokinase enzyme. Then in the second step, we are converting glucose 6-phosphate into fructose 6-phosphate. We are not adding here. We are not adding here something. We are not deleting here. This is just a isomer step. Then which enzyme we will use? We are making an isomer. Enzyme should be isomerase. Enzyme should be isomerase. And then fructose 6-phosphate, we are getting converted fructose 6-phosphate into fructose 1,6-bisphosphate. Again, we are breaking down a ATP. So the enzyme name is, this is again very important. नीट में ये भी काफी बार क्वेश्चंस आ चुका है. I'm deleting it here. Are you making this table in your notes?
Yes, miss.
So enzyme name is phosphofructokinase. We will remember these three enzymes. The first one is hexokinase, second one is we have isomerase, and the third one we have phosphofructokinase. And in the next step, we are converting fructose 1,6-bisphosphate into triose phosphate. Here we will need an enzyme which is known as aldolase. Enzyme name is aldolase. ठीक है, repeat करो फिर से. The first one we have glucose, then glucose से we have made glucose 6-phosphate, then fructose 6-phosphate, fructose 1,6-bisphosphate, and then triose phosphate. Are you able to learn first 5 steps?
Yes, ma'am.
Yes, ma'am.
After converting the triose phosphate, we are converting it into 1,3-diphosphoglyceric acid. It means we are adding here 2-phosphate now.
Yes, ma'am.
Triose phosphate, and then we are converting it into 1,3-diphosphoglycerate, or we can also call it diphosphoglyceric acid. In this step, this is very important step. In this step, we are making 2-NADH. Do you know what is NADH? NADH is also an energy currency. NADH is also energy currency. Like ATP is in the form of cash, and NADH is like a checkbook. If you have a check, then you go to the bank and then give it to them, and they will give you a cash. NADH is just like this. We have a check. Okay? So how many NADH we will make here? 2 NADH. In the first step and in the third step, we are consuming 2 molecules of ATP. But at 5th step, we are synthesizing 2 NADH. NADH will form from here NAD positive. Let's write it down in the form of this. We have a NAD positive, and I am adding here H positive, then it will make NADH. What type of reaction is this? Addition of hydrogen is? Addition of hydrogen is? Reduction. Reduction ना? तो यहाँ पे NAD positive will act as a which agent? कौन से एजेंट के जैसे बिहेव करेगा?
Reducing agent.
Reducing agent or oxidizing agent?
Oxidizing agent.
Oxidizing agent. So here NAD positive will be have as which type of agent?
Oxidating.
Oxidizing agent. This question is already asked many times. Oxidizing agent. NAD positive will behave as a oxidizing agent. If we need to make 2 NADH, then how many H positive do we require?
Two H.
Obviously, 2H positive. So we will call it, we need 2 redox potential. We need 2 redox potential. 2 redox potential for addition of 1 H positive. 1 H positive required how many redox potential? 2 redox potential. So if I ask for the addition of 2H molecule, how many redox potential required? Four redox potential. Simple.
Yes, ma'am.
Okay, so let's quickly summarize it. Firstly, we are converting triose phosphate into 1,3-diphosphoglyceric acid. In the conversion of this step, we will make 2 NADH by the help of an oxidizing agent, which is your NAD positive. Then 1,3-diphosphoglyceric acid will get converted into 3-phosphoglyceric acid. Again at this step, again at this step, we will make 2 molecules of ATP. How many molecules? We will make 2 molecules of it. Okay? Then at the last step, where we will convert 2 phosphoenol pyruvate to pyruvic acid. In this step also we are making 2 ATP molecule. So if I ask what is the total gain of ATP here? Only ATP. I'm just asking ATP only. 2 ATP here, 2 ATP here.
Four.
4.
And in the initial step, we have consumed 2 ATP minus those 2 ATP.
2.
2 ATP. So in the entire process of glycolysis, how many gain of ATP is there? 2 ATP.
2
And if someone asks total energy, then we will say 2 ATP plus 2 NADH. 2 ATP plus 2 NADH. Done? Maximum question is already asked from this table only. Like at which step we use 2 ATPs. In the first step, we have used glucose to glucose 6 phosphate, and the second ATP we have used in glucose 6 phosphate to glucose 1,6 bisphosphate. Second question can be asked, at which step we gain first ATP? At which step we have gained first ATP? Which step is this?
Glucose.
At which step we have gained first ATP? Our first ATP, like our first सैलरी होता है ना जॉब के बाद, तो फर्स्ट सैलरी फर्स्ट ही रहता है हमेशा. ऐसे ही इस पूरे स्टेप पे सबसे पहले मेरे को डायरेक्ट एटीपी किस स्टेप में गेन हुआ है? इन द कन्वर्जन ऑफ 1,3-diphosphate to 3-phosphate. This is my first gain ना.
Yes,
Yes,
ma'am.
ma'am.
First gain है ये मेरा. So this step can be called as SLP step. SLP. What do you understand by SLP? First substrate level phosphorylation. So हमारा SLP क्या होगा? What do you understand by SLP? SLP is that step at which we will gain first ATP, our first ATP. So the total gain of glycolysis is 2 ATP. We can write it here now. We'll just write it here. We'll gain here 2 ATP with 2 NADH. Are we done with the first step, glucose to 2-pyruvic acid?
Yes, ma'am.
We have made here 2 molecules of pyruvate. Is how many carbon compound?
3 carbon.
3 carbon compound. Very well done. Chal. The next step is conversion of pyruvic acid into Can anyone tell me the formula of pyruvic acid? This is CH3 C double bond O COOH. This is your pyruvic acid. Okay. Now we will convert this pyruvic acid into acetyl CoA. So by the conversion of it into acetyl CoA, will be needed a 2 coenzyme A. We have 2 pyruvic acid. We'll write it here 2. And we will also need a oxidizing agent. Which is your oxidizing agent?
NAD.
NAD positive. Okay. Then it will get converted into 2 molecules of acetyl CoA. Pyruvic acid is a 3 carbon compound and acetyl CoA is 2 carbon compound. Where is 1 carbon? एक कार्बन किधर गया हमारा? बेसिकली 2 ना, 2 पायरुविक एसिड से 2 एसिटाइल CoA बना, तो 2 कार्बन कहाँ गया? 2 कार्बन lost in the form of CO2. 2 carbon lost in the form of CO2, and these NAD positive get converted into NADH, 2 molecules of NADH. So this is your link reaction. What is the gain of total? What is the total gain of link reaction? 2 NADH plus 2 CO2. We can write only 2 NADH here. There is a role of one enzyme. There is a role of one enzyme. The enzyme name is pyruvate decarboxylase and pyruvate dehydrogenase. Very important. Pyruvate decarboxylase. D means removal. Carboxylase means CO2. Pyruvate dehydrogenase. Dehydrogenase means removal of hydrogens. Done. NAD positive will behave here as again oxidizing agent. So after link reaction, acetyl CoA will enter into
Oxaloacetic acid.
Krebs cycle. Okay, so I'll tell you a most interesting trick to learn entire Krebs cycle. Let's have a look here. A city is kept silent for military operations. ठीक है? A signifies here acetyl CoA. City, citric acid, or we can call it citrate also, is isocitrate. Capped alpha keto glutarate. Silent signifies here the first one we will have an enzyme succinyl CoA, and S also signifies here succinate. 4 represent fumarate, military, malate, operation, oxaloacetate. याद करो उसको, just try to remember it. A city is kept silent for military operation. A acetyl CoA. Acetyl CoA will combine with, because this is a cycle. So acetyl CoA will get combined with oxaloacetate and it will form citrate. Then citrate will get converted into isocitrate, which is a isomerization process. Isocitrate will get converted into alpha ketoglutarate. Alpha ketoglutarate will get converted into succinyl CoA. Succinyl CoA will get converted into succinate. Succinate will get converted into fumarate. Fumarate will get converted into malate. Malate will get converted into oxaloacetate. Is it simple?
Yes, ma'am.
Everyone, सुन रहे हो? क्लास में सो गए हो?
Yes, ma'am.
चलो फिर. If you are done with this, now we can draw our cycle. Now I'll ask with you only. Tell me the cycle. Firstly, we have acetyl CoA. Acetyl CoA will combine with
Oxaloacetate.
oxaloacetate. So it will get combined into oxaloacetate and it will form.
Citric acid.
Chalo, we'll write it citric acid. After citric acid,
Isocitrate. Isocitrate.
and then isocitrate will get converted into.
2-ketoglutarate.
That's not 2. This is alpha.
Alpha
Alpha.
ketoglutarate.
And alpha ketoglutarate will get converted into
59.
succinyl CoA
CO.
in the form of enzyme. तो सबसे पहले enzyme आता है. After that we get a product ना. What is our product? Very good, Harshada. Succinate. And succinate will get converted into
Fumarate.
fumarate. Then
Malate.
Malate.
then
Oxaloacetate.
oxaloacetate. Simple. तो इसको हम थोड़ा सा इधर लिख देते हैं. We'll write it here. ठीक है? Now we are done with our basic cycle and now we will write its components. Acetyl CoA is a how many carbon compound?
2.
2 carbon. Very good. And citric acid is a 6 carbon. 4 carbon compound कहाँ से आया यहाँ पे? Oxaloacetate. 4 carbon oxaloacetate get combined with 2 carbon acetyl CoA, then we will get 6 carbon citric acid. Isocitrate is also 6 carbon, but alpha ketoglutarate is 5 carbon. Where 1 carbon goes?
Carbon dioxide.
Yeah, it get released in the form of CO2, and CO2 will not release alone. It will release 1 molecule of NADH also. ठीक है? Next, succinyl CoA is again 4 carbon. Where 1 carbon left? 5 carbon to 4 carbon. Again, 1 molecule of CO2 remove, and it will not remove alone. It will be with NADH. ठीक है? Succinyl CoA will get converted into succinate. तो here is something same ना? Succinyl से succinate बन रहा है. And we all know ATP is equals to GTP. So we will get our ATP का same molecule. We will get similar to ATP. That's your GTP. We will get 1 molecule of GTP here. And then succinate will get converted into fumarate. We will get something in the form of F. So we get NFADH2. And at this step, we will get nothing. Then after converting malate into oxaloacetate, we will get again NADH2. This is your diluted NADH2. Simple. Let's recap it again. This is our Krebs cycle. Firstly, we have acetyl CoA. Acetyl CoA, we will get citric acid with the help of oxaloacetate. Then citric acid will get converted into isocitrate. Isocitrate will give rise to alpha ketoglutarate. Alpha ketoglutarate is a very crucial step. because you are just looking at here कि इस step से just before this step and just after this step we are getting one molecule of CO2. It means in totality we have two molecules of CO2 and also two molecules of NADH2. So alpha ketoglutarate is a very crucial step. By converting succinyl to succinate, we get something similar. ATP is similar, we have GTP. So we will get here GTP. Then after conversion of succinate to fumarate, we will get something in the form of F. So we get FADH2. FADH2 is also energy currency. ये भी एक energy currency है. Then in the case of malate, we will get nothing. Malate से conversion में oxaloacetate, we will get NADH2. And are you all done with this step? Malate is again 4 carbon. You can write it here. This is 4 carbon. This is again 4 carbon. This is again 4 carbon. And oxaloacetate is already 4 carbon. Done, बच्चों?
Yes, ma'am.
Yes, ma'am.
Complicated लग रहा है थोड़ा सा?
Yes, ma'am.
कोई नहीं, एक दो बार पढ़ोगे ना, then you feel it very simple. चलो, tell me what is the total gain of Krebs cycle? We got 2 CO2. We got 1 GTP. We got
1 FADH2.
3 NADH2 and 1 FADH2. This is the gain of one cycle. We have this gain is just from one cycle. And just remember, we have 2 acetyl CoA now. We have 2 acetyl CoA. Look at here. We have 2 acetyl CoA. So we need to run this Krebs cycle how many times?
2 times.
2 times. So we will multiply all these things by 2. 2 से मल्टीप्लाई करेंगे हम लोग इधर इसको.
Yes, ma'am.
Very good. So चलो, we'll write our total gain now. Let's have a total gain. Total gain is important. Glycolysis किधर हो रहा है हमारा? It is happening in cytoplasm. How many ATP we get here? 2 ATP with 2 NADH. In the link reaction, we got 2 CO2 plus 2 NADH2. Write it 2 NADH2. And in the Krebs cycle, what is our total gain?
4 CO2.
Very good. 2 GTP, 2 FADH2.
6 NADH2.
Very good. 6 NADH2. Can you calculate the total ATP from here? Just telling you, देखो. 1 NADH is equals to how many ATP? 3 ATP. 1 GTP is equals to how many ATP? 1 ATP. 1 FADH2 is equals to how many ATP?
2
2
ATP.
ATP. Chalo. Try to calculate the total ATP. Then we'll solve some questions. Do it. Yeah. You can type your answer in the chat section also so that I can get your names also. Which students is solving right, so student is doing some mistake. Okay, Harshada, you're seeing 21 ATP. Harshada is now at 24. Okay. ऊपर जो मैंने बोला ना 1 NADH is equals to 3 ATP, उसको पट करके निकालना है. It means you can look at here कि 2 NADH2 है we have, so it means 2 NADH2 is equals to 6 ATP. Here it is also 6 ATP and 6 NADH2 6 3 ja. It means we have 18 ATP. Then FADH is equals to 2 ATP. So we have
24 ATP.
2 4 ATP and ये तो 2 ATP है. So we'll calculate 6 plus 2 8 8 plus 6 14 14 plus 2 16 16 plus 4 20 20 plus 18 38. Are we getting 38? No one is getting
Yes, ma'am.
38. Should I repeat?
Ma'am, I got 38.
Very good, Gayatri. Let's repeat it. We have 2 ATP here. Okay? We have 2 NADH2. 1 NADH is equals to 3 ATP. Then 2 NADH is equals to how many ATP? 6 ATP. Then again we have 2 NADH. 2 NADH is equals to 6 ATP. 2 GTP. 2 GTP is equals to again 2 ATP. 2 FADH. 1 FADH is equals to 2 ATP. So we will write it here 4 ATP. 6 NADH is equals to how many ATP? 6 3 ja 18 ATP. Let's count it. 6 plus 2 8. 8 plus 6 14. 14 plus 4 14 plus 2 कर लो पहले 16 16 plus 4 20 and 20 plus 18. I told only of nahi beta, you just need to add all over the game. Very good, very good. Chalo, let's solve some questions. Conversion of glucose to glucose 6-phosphate is the first reversible reaction is catalyzed by which enzyme?
C. Hexokinase.
Very good. W ना PYQs में आता है ये question, so it has already asked in NEET 2019. The correct is C. Next question. What is the role of NAD positive in cellular respiration? Yes, Mishba.
Ma'am, option C.
Option C. It is a nucleotide source for ATP synthesis. Is it correct? Are we going to synthesize any nucleotide from NAD positive? We were just writing it here. कि NAD positive is behaving as an oxidizing agent. NAD positive behaving as an oxidizing agent. Then why we are writing it here nucleotide source?
It functions as an electron carrier of the
Exactly. Very good, Anu. It is functioning as an electron carrier. It is carrying electrons. Gain of electron is reduction. That's why it is behaving as oxidizing agent. Loss of electron is oxidation. Gain of electron is reduction. The correct is B. Function as an electron carrier. Done, bachcho?
Yes, ma'am.
Next question. What is the link between glycolysis and TCA cycle is also known as
Krebs cycle.
Krebs cycle. Link बताना है.
Option B.
Everyone. Anushka is saying C. Shikha is saying B. Okay. Rehana, why are you late? Very good. This is very simple. The correct is acetyl-CoA. Link reaction, that why we were calling it. Next, respiration is which type of process? Energy taking or energy giving? Catabolic or anabolic?
Catabolic.
Catabolic. So we can ignore our anabolic steps. Just find it out. Is it endergonic or exergonic? Endergonic. Energy consuming. Exergonic. Yeah, exactly. Because it is giving us energy in the form of ATP. That's why this is exergonic reaction. Next question. Acceptor of acetyl CoA Krebs cycle is
Option D, oxaloacetic acid.
very good. This is oxaloacetic acid. Are you all clear with this point? Gayatri, not C beta. Acetyl CoA is getting combined with oxaloacetic acid and then we are gaining citric acid.
Sorry, ma'am, I don't clearly see.
No worries, no worries. It happens sometimes. Okay, then we are just done with the question practice. Let's summarize today's learning. What we have learned today, we have learned the mechanism of glucose, how glucose is getting converted, how we are taking exactly energy from the glucose. We have 3 steps involved here. The first is
Glycolysis.
glycolysis is a how many step process?
TCA cycle,
10
electron
step
pair
process.
transfer reaction.
10 step.
10 step, very good. Then we have link reaction and then we have
Krebs cycle.
Krebs
Krebs
cycle.
Krebs
cycle.
cycle. Krebs cycle is also represented as TCA cycle. What is the total gain of all 3 cycles?
38.
38
38
ATP.
in case of
38
aerobic reaction. In case of aerobic
Aerobic
respiration.
reaction.
ठीक है? Glycolysis is happening where? In the cytoplasm. And these two are
Mitochondria.
Mitochondria.
matrix of mitochondria. These two are carrying out an matrix of mitochondria. I just forgot to tell you one major point. Let's go back to our slide. Yeah. Just write it down here. In the conversion of succinate to fumarate, there is an involvement of one enzyme. That enzyme is known as succinate dehydrogenase. And this enzyme is very important. Why it is important? Because succinate dehydrogenase enzyme found in inner membrane of mitochondria. ये आपका माइटोकॉन्ड्रिया का इनर मेम्ब्रेन में पाया जाता है एंड दिस एंजाइम is getting transferring is getting transporting itself from inner membrane to matrix just for carrying out this reaction. So it means this enzyme is very crucial now. And this question is several times asked in the NEET. Which enzyme is very important for Krebs cycle? Like which enzyme of Krebs cycle is in the inner membrane of mitochondria? This is your succinate dehydration enzyme. And if I ask you, which one will be the SLP step in this reaction? SLP step. First time energy gain. Direct energy gain. कौन सा होगा SLP?
Oxaloacetic acid.
Between oxalate and malate.
गैलो एसिटेट में कहा फर्स्ट टाइम में हमारा जो फर्स्ट प्रोडक्ट है ना वो सिट्रिक एसिड है. Acetyl CoA is getting is combining with oxaloacetate. It is converting into citric acid. This is our first step. This is second step. This is third step. This is fourth step. This is fifth, sixth, seventh, eighth. So at which step we are getting
Third step.
first anode? No, at the first step we are getting it in the form of NADH2.
Fifth step,
Yeah, very good, Anu. At fifth step, we can call it
because
first
GPP is
SLP.
equal to
Huh.
ATP.
Equals to it. We are getting here direct energy. This is our first SLP step. These are again two major important points. Okay? So we have finished our major learning here. I have already shared a module with you in your group. Did you try to open the PDF?
Yes, ma'am.
If you
Yes,
will
ma'am.
open, you will get to detailed notes with questions also. So try to solve all those questions before the next class. Then you'll get better understanding of this chapter. And in the next chapter, we'll start with, we have already finished Kelvin cycle. In the next class, we will start with anaerobic,
Respiration.
and then we will start ETS and RQ. We will finish this chapter in next class.
मिस मेरी प्रेजेंट ही लग गई ना।
हाँ बेटा लग गया आपका।
मिस मैं लेट ज्वाइन हुई थी, मेरा कॉलेज लेट छूटा था इसलिए।
No worries, आपका अटेंडेंस जैसे ही आप एंटर हुआ लिंक के थ्रू, यू हैव एंटर्ड योर करेक्ट मोबाइल नंबर ना?
यस मिस।
Then your attendance has been already marked. So we'll start with anaerobic reaction, ETS and RQ. So try to just have a look with all these topics before coming to the class so that we can finish this chapter in the next class. And after finishing this chapter, we'll start with photosynthesis in plant, and then we'll go on human physiology. Okay? Okay then. We'll see you in the next class.